Hi, Brittany.
Yay, how are you?
I'm good. I'm good.
Raise me. Okay, I'm trying to see where I can share my presentation.
Uh, Down, sharing screen.
I don't have that ability. All I can do is chat, react, raise my hand, or do the Q&A.
Okay.
He signed in as an attendee instead of a panelist.
Ah,
Yeah,
there
I
we go.
just did.
Okay.
I
If you
just
have
Oh,
did.
signed in as a panelist, you would have received a panelist email.
Perfect. We can see your presentation. Perfect. Ready to start, Brittany? Hello, can you hear me?
I can hear you, Neza, but I'm not hearing Brittany.
Yeah, maybe.
oh, there we go. Now is that better?
Yes, we can hear you now.
That's good. There we
So
go.
ready to start?
Yes, I am ready to start. Sorry about that.
Okay, perfect. We have 6 people who joined. Do you wanna us to wait for a few minutes or just start and accept people?
It's completely up to you all. I'm only on a single monitor right now, so I can only see my presentation. I can't see everybody, but no, it's completely up to you guys. If you want me to start, I will go ahead and start. Um, yeah.
I think we will just start and then people will join progressively.
Yeah, that sounds perfect. Okay, so
Okay,
just, oh, sorry. Yep, you go first. My bad.
I will introduce you to people who already are here. So, good morning, everybody, and thank you for joining us today. We are pleased to have you joining us in this insightful presentation on addiction neuroscience. So we welcome Dr. Brittany Kuhn, which is an assistant research professor at the Medical University of South Carolina, US. Dr. Kern has extensive expertise in neuroscience of addiction. In fact, after completing her PhD at the University of Michigan, studying corticothalamic circuitry in addiction, she joined Dr. Peter Kalaiva's lab at MUSC. Her work focuses on the ventral pallidum's role in opioid addiction, vulnerability, using translational rodent model. She is using rats and mice. She recently received a grant to establish her independent research program examining circuit level mechanism underlying individual differences in addiction susceptibilities. Today, she will present her latest finding on ventral pallidum regulation on heroin addiction susceptibility. Following Dr. Kern's presentation, we will have a 15-minute question and answer session for discussion. I encourage you to prepare and write your question on the question and answer session below, and we will address them after Dr. Kern's talk. So without further delay, I'm pleased to turn the floor over to Dr. Kern. Thank you.
Thank you, Neza. I appreciate it. So thank you everybody for the invitation to talk to you today. This is the first time I'm going to give this talk in the way I'm giving it. So hopefully it goes well. That being said, I am only using one monitor right now. So if anyone has any questions at any point, or any clarifications you need, I am not the person who will be offended if you interrupt me. So just interrupt me. But today, what I'm going to talk to you about is the role of the ventral pallidum in regulating heroin addiction susceptibility. So I'm going to really look at this from two different perspectives. As Neza mentioned, I do work using both mouse as well as rat animal modeling. So I'm going to start off by looking more just at neurobiologically, how is the ventral pallidum contributing to different heroin-seeking behaviors. And then I'm going to bring it into looking at it within the perspective of a model that naturally captures individual variation to opioid addiction behavior. So something that's a little more translationally relevant to the human condition. All right, so to get us started, I just want to orient everyone to just substance use disorder, or SUD. So substance use disorder is characterized as a chronic neuropsychiatric disorder whereby compulsive drug use and relapse occur despite attempts to remain abstinent. And that's really important to take note of. I'm sorry, this is actually data in reference to the United States, so I apologize, but at least within the United States, we see that about $700 billion a year is cost to the economy when it comes to substance use disorder. Given the prevalence worldwide of SUDs, I'd imagine it's relatively representative of other nations as well. What we see, though, that's really interesting with substance use disorders and a little different from other psychiatric disorders is that there is an increase in SUD prevalence, but it's occurring across multiple communities. So within a specific drug, we may see that it's affecting more, females versus males, one race versus another. But overall, the increase that's occurring is happening across multiple communities. So what this is really telling us is that this is something we need to address, and it's something where we need to dive a little deeper and start to really understand, why is this happening on a global scale? Why are we seeing this big push? And one of the ways we can really start to look at this is within the United States, but this is coming from the National Institute on Drug Abuse. And what we're seeing here is a 20-year span and the number of drug addiction related overdose deaths. And what I want you to really take note of is that really within the last about 10 years or so, we're seeing this really big increase in different opioids. So we have our synthetic opioids right here. We have our heroin in this gray. And so heroin is actually the drug that I use. And then we also have prescription opioids, which seem to just kind of been increasing slowly over 20 years. So I think just to, be blatant, we're all aware of the fact that there is a big opioid epidemic that's happening worldwide right now. So obviously that is being reflected here in these numbers right here. But what this really tells us is the fact that opioids are something that we really need to focus on right now. That is a very big health concern across our communities. But what's really interesting when you study substance use disorder is that not every single person who tries a drug or even recreationally uses it to a certain extent is going to become an addict or is actually going to be diagnosed with a substance use disorder. So to put this in perspective, and again, I'm sorry, this is data looking at an American population, but if we look at the adult American population within the United States, which seems to be about roughly 220 million, what we see is that only 8.4% of the adult population is actually being diagnosed with a substance use disorder. So what does this tell us? I think we can all appreciate the fact that more than 8.4% of our population globally is engaging with either an illicit or an illegal substance to some extent, alcohol, tobacco, marijuana in certain areas, and then the illicit substances. But only 8.4% of the population is actually getting a diagnosis. So what this is really telling us is that there is a lot of variability in what is ultimately conferring vulnerability or resiliency. And there's a couple different ways we can tackle this question or really look at it from different perspectives. And I'm going to show you two different perspectives today. The first is to start to dive directly into the neurobiology. Could this variation be that there's different neurobiological mechanisms that are pushing one direction or another? Or, and what I'll show you later, is it a function of just behavioral variability? Do we need to instead start to look at these different behaviors and this different neurobiology within the perspective of a model that captures behavioral variability? So let's start out with the neurobiology. And what I'm going to show you on this next slide is a very kind of reductionist perspective on the neurobiology of substance use disorder. All right, I don't want you all to get lost in the weeds of looking at what all the legend means and everything up here. What I just want you to appreciate is we are seeing, and this is in a rodent brain, that there's a lot of interconnectivity between cortical, subcortical, and brainstem regions that are contributing to the pathology of substance use disorder. This is not as something that is just occurring within one area region of the brain. It's something that is globally is bringing in multiple areas of the brain. And the area that I'm going to focus on today is the ventral pallidum. So the VP is right here in rodent brains as well as in humans. The ventral pallidum sits right below the anterior commissure, so very much more of a subcortical area. One region that we know people love to talk about when it comes to addiction is the nucleus accumbens. You can see it's in very close proximity to the ventral pallidum. The nucleus accumbens is a wonderful structure. Addiction, people love it. It's great. We are not going to talk about it basically at all today, though. So the ventral pallidum, why am I so interested in the ventral pallidum? What's happening here? Especially because you can see that there's other structures like the nucleus accumbens or the VTA or the amygdala areas that are getting a lot of attention in this schematic. So why am I interested in this little nuclei that it really seems like everything's skirting around it? Well, let's dive into that. So the ventral pallidum is the main output structure of the nucleus accumbens. And so because of that, for the longest time, the VP was really looked at as being a motor relay station. So not being able to actually mediate any motivated behaviors on its own. Until about the last 2 decades, where some really beautiful work has come out that has shown that the VP can independently mediate motivated behaviors, and that actually sometimes that these neural signatures are preceding what we see in the nucleus accumbens. So that really starts to bring into question of who's really the master orchestrator here. Is it the nucleus accumbens or could it be the ventral pallidum? But regardless, we do see that the VP is very important for mediating motivated states. What about for addiction? That's what I'm really interested in, right? What's happening in the VP in addiction? So we've seen more recently, so this kind of more in the last 10 years, a lot of beautiful work showing that drug-seeking behavior for alcohol, cocaine, and more recently, one little opioid study showing that the ventral pallidum is an important structure for seeking behavior. And ultimately, seeking behavior is what we use to look at relapse behavior. So that's really important to know. Let's learn a little bit more about the ventral pallidum though, right? So it's always nice to know that a structure is involved, but let's start to get a little bit more mechanistic. What about the ventral pallidum is contributing to drug-seeking behavior and how is it contributing? So one way we can answer this question is looking at the cellular composition of the ventral pallidum. So the VP is composed really of 4 main subtypes. We have GABAergic cells, which make up about 80% of the population. Within GABAergic cells, we also have a subset that express and co-release enkephalin. Enkephalin is a neuropeptide. We then have about 10% of the population, which is glutamatergic. Another 10% is cholinergic. And then we start to see this group of parvalbumin cells. So parvalbumin cells are, generally speaking, fast spiking interneurons. But we see in the ventral pallidum what's really unique about it is that parvalbumin cells are encasing both GABA as well as glutamatergic cells. All right, so we see these different cellular differences. Why do we care? The reason we care is because there's a really beautiful paper that came out in 2018 that was really the seminal work from Lauren Faget in NASCO's lab at UCSD that showed we very much should care about what's happening with these cellular phenotypes. Because guess what? These GABA cells really seem to be acting to reinforce or increase seeking behavior. But these glutamatergic cells, this little population, it's only 10%, is what is mediating aversive behaviors. So this is really fascinating. This is like what we see in areas like the nucleus accumbens, where we seem to have this push-pull mechanism. Within the exact same structure, we have cellular phenotypes that are doing the exact opposite things as one another. So my interest was, let's dive a little deeper, let's learn a little bit more about this. And what really inspired my work was 2 different, really one specific piece of work. which came from my boss's lab. This was before I joined. This is a prior graduate student, Jasper Heinsbrook. He's now faculty at UAB down in Alabama in the United States. He did really beautiful work where he wanted to say, okay, I understand that GABA and glutamate for a natural reward are doing two very different things. Drugs of abuse create a pathological state. It creates a difference in neurobiology. What is happening now if instead of a natural reward, we make it a drug reward? Are we maintaining this relationship, or does that relationship somehow go awry? And what he found is that if cocaine was used as the reward, if he activated GABA cells, he showed that he could increase seeking behavior when there were no drug paired cues present, but not when there were drug paired cues present. So a little interesting. If he activated enkephalin cells, which is a very small subpopulation of GABA cells, he found that he could just increase seeking behavior both in the absence and the presence of these cocaine paired cues. And then if he activated glutamate cells, he could attenuate this seeking behavior. So really what he showed was pretty similar to what we saw for the natural reward, right? If you activate the GABAergic cells, we're seeing an increase. If you activate glutamatergic, we're seeing a decrease. He sees some really interesting nuanced differences of if the GABA cells contain enkephalin or not, which is really interesting, that I'll go into a bit later. And then this really inspired the work that I started doing. But over the course of doing my work, this also really beautiful paper came out. And this is the only paper that's looking at cell-specific VP regulation as it comes to opioids. And this came out in 2022 from Steve Mahler's lab at UC Irvine. And what he essentially found, they only focused on GABA here, but it's very similar to cocaine. If you activate GABA, you're going to increase seeking behavior. You can see they did two different types of seeking behaviors, but you increase seeking behavior. And then if you do the opposite and you inactivate it, you decrease the seeking behavior. Now, they didn't look at glutamatergic population at all because this work was actually done in mice. This was done in rats, and we don't have the ability in rats to really cleanly look at glutamatergic cell manipulation. But what they did do is take it a step further and say, is this cell population necessary for other behaviors in substance use disorder? And so their main question was, does it affect consumption of remifentanil? So when we're doing that taking phase, is if we manipulate GABA cells, is that going to affect anything? And the answer was no. So it does seem like these specific cell populations are tied to a seeking state, whether that aberrant seeking is occurring with or without cues. So this is where my question really came in, is now let's look at this cell-specific regulation in the context of heroin seeking, and let's look at it both in the absence and the presence of heroin. So very much looking at what Jasper's 2020 paper was, but now instead I'm going to be using heroin as opposed to cocaine. So before I dive a little deeper, I always like to give everyone anchoring points of what I'm going to talk about today. So I'm just going to show you some of the modeling I've been doing, looking at VP cell-specific regulation. I'm going to show you behavior for 3 different cellular phenotypes. So that GABA, that 80% of the population, enkephalin, which is about 10% of this population, and glutamate, which is 10% of the overall population. I'm going to show you data where we stimulated these cells, and then also some data that we're really excited that we're going to be sending out for publication soon from a visiting faculty who's actually, I think, on a plane right now back home to Cameroon. So Dr. Bryce Agisi-Owana. So I don't know if he'll actually be able to watch it, but we're giving his data a little shout out. Then I'm going to dive a little deeper and I'm going to go into mechanisms. Whenever we see differences, the next big question in mind is, what is the mechanism? So if we see cellular differences here in what's mediating seeking versus attenuating seeking behavior, what's driving that behavior? What pathways are really mediating that behavior? And then we're going to take more of kind of like this theoretical or ideological step back and really start to look at how do VP differences fit into a more global translational model of addiction. All right, so let's dive into it. So for this first half of the talk, I am going to be focusing on work using transgenic animals. I use mouse modeling here. So this is my experimental paradigm that I'm going to be using. So what I do is I use animals that selectively express CRE in either GABA, enkephalin, or glutamate cells. And because they selectively express Cre, I'm then able to use a Cre-dependent stimulatory dread and inject that into the ventral pallidum. And that is what is allowing me to selectively activate just one of those 3 cell populations within the VP. I activate it by giving the animal systemic injection of CNO, and then saline is just a control. So I do the surgeries on my animals. I give them the injections to express that stimulatory dread, as well as catheter surgery so that the animal can self-administer the drug. And then after a period of rest, the animals go into self-administration. So I do 12 sessions with 1 day break in between the 6 sessions, and I do 3-hour sessions per day. So what we see here is that we have, this is actually, I think, a rat, but we're gonna just pretend it's a mouse right now. So I have a mouse, and when the mouse goes ahead and engages, we actually use ports, but these are levers. On the active lever, we see that we get the presentation of both a tone and a cue light, and the animal gets the heroin infusion. If they press the inactive, the animal doesn't get anything. So over the course of this, the animal starts to learn that if they preferentially or selectively press this lever, cue presentation and heroin infusion. The animals then have a period of forced abstinence where we just let them hang out in the colony room and just let them relax, followed by extinction training. So during extinction training, what you can now see is that the animal presses on this active lever, no cue presentation, no heroin infusion. So now they're learning something new. They're learning that if they did what they previously did, they're no longer getting the reward or the reward paired cues. And then the very end, I do test for cue-induced reinstatement. So now when the animal presses on the lever, we see we get the drug-paired cues, but not the drug itself. So now we're asking the question of, well, when those cues that were associated with the drug delivery are present, how does that motivate seeking behavior? So I do 2 different testing periods here. You can see in this light blue. I ask the question when we're in late extinction training, so these animals have been in extinction for at least 9 days. If I activate the cell population, is that going to affect any type of seeking behavior? So are we just going to generally affect seeking behavior that's not tied to a cue or anything, just the general behavior? Whereas during cued reinstatement, I'm asking the question of when we now specifically have the cues present, how is that affecting behavior? So 2 different ways of looking at seeking. And you'll notice that I have 4 little dots. We did a within study session. So each animal received CNO on 1 day and saline the other day for both of these tests. And we did counterbalance everything so there wasn't an order effect. All right, so let's see what the data looks like. All right, so this is the only time I'm going to show you kind of the entire pipeline in terms of the behaviors. But I just want you to have one time where you can just really see how the modeling looks. So I have my GABA animals on top that selectively have the stimulatory dread in the GABA cells. Enkephalin, where we selectively have that stimulatory dread in the enkephalin cells. So what you're seeing first is looking at their self-administration behavior, which is in the colored block, and then the extinction behavior, which is just in white. So really what you're seeing is that animals are increasing active presses, which is what we have in our dark black. Our light gray is inactive, and our little triangles that are yellow are infusions. So overall, animals are discriminating between the 2. They're earning infusions, and during extinction training, they decrease their seeking behavior. So that's what you want to see. If you do rat work, this looks significantly less pretty than rats. Mice are very, very variable little guys, so their data will never look as nice as rats, but you know what makes them more fun to deal with? And so the first test that I did, I told you about, was that extinction test. During late extinction, if we turn on the GABA cells or the enkephalin cells, what happens? Nothing. So we can see that there's no difference between giving vehicle or CNO in either of these populations. This is interesting because for cocaine, we saw an effect here. All right, so if we move on, now we're going to get to the point where we want to look at cued reinstatement. So when we look at cue-induced reinstatement, we want to make sure that the animals actually showed relapse behavior. So what I'm showing you here is the active port and the inactive port. This is going to be the last day of extinction versus their vehicle treatment versus their CNO treatment. So really what we want to see is a difference here. And we see that for both the vehicle and the CNO, we have a difference, but please take note of the difference in the axes labels. So to look at this a little better, what we see is that if we selectively turn on the GABA cells or the enkephalin cells during cued reinstatement, we see this marked increase in seeking behavior. So again, this is what we saw for cocaine. This is a little different. In cocaine, we saw that turning on GABA did not affect cued seeking behavior, but in heroin it is. So we are seeing some nuanced differences between different types of drug classes and how the VP cell types are mediating it. And then if we just are good little scientists and we do our CNO control to make sure that CNO alone isn't doing anything to behavior, we see it's not. So this is really great. This is nice. This aligns kind of nicely with the remifentanil data and also shows some nice, beautiful differences as well as complementing the cocaine data. But let's look at glutamatergic cells. What are glutamate cells in the VP doing during heroin seeking? Because nobody has looked at this yet. So we have the same thing here. We have our self-administration followed by our extinction. The animals are differentiating. They're looking good. But when we do that extinction test, now we see that turning on the glutamate cells is decreasing that seeking behavior. So just this aberrant seeking behavior that's happening at the end of extinction training, we're able to attenuate that. If we look at our cued reinstatement, this is great. We see what we want to see. When the animals are just given vehicle, so we're not manipulating anything, they're reinstating, they're relapsing, they're showing more active lever presses. But when we give CNO, what's happening here? This is an incredibly drastic decrease in drug-seeking behavior, and this was really exciting to us because what we see is when the animals have vehicle, we show nice reinstatement. CNO, they almost all plummet just straight to zero. The reason this is such a really exciting result is remember, this glutamatergic population is only 10% of the VP population. So activating 10% of a population is having an unbelievably potent effect on their behavior. So that was really exciting to us. That opens up a really beautiful window of further trying to see what exactly are these glutamate cells doing? How are they engaging maybe with other cells in the VP, with other circuitry, other mechanisms, what could be going on here? And then again, we're being good scientists and doing our CNO control. And as expected, we see that CNO alone doesn't do anything to behavior. So that's really great. So the take-home message here is that it does appear that these VP GABA and enkephalin cells are just mediating maybe more the motivational properties that are associated with the heroin-paired cues. When we manipulate those cells, when we stimulate them, we only see effect during cued reinstatement. So it seems that these particular cell types are tied to something that's associated with those particular cues. However, it looks like the glutamatergic cells may be acting as just a general brake on behavior. So it doesn't matter if there's reward paired cues there or not. If you have any type of aberrant seeking happening, this is acting as a brake to shut this down. But then let's look at the opposite. What happens if we inhibit this population? And what's really important about looking at this is that when we're stimulating a population, we're artificially imposing a situation on that region or that animal. When we're inhibiting something, we're allowing systems to act as they normally would, and we're taking something offline. So if something was never online, theoretically speaking, we shouldn't be seeing much of an effect. But if something is online and we remove it, what's happening? And this was the work that that Bryce did, and he did a really beautiful job. He accomplished this test in a year, so he should be very proud of himself. And so what I'm going to show you here is a little bit of a different perspective. I'm going to show you the phenotypes, but I'm going to show you their extinction day, vehicle, and CNO. So this is only during cued reinstatement. So what we see is that when the animals had cued reinstatement where they got vehicle, they showed increased seeking behavior. That's beautiful. That's what we want to see. But if we inhibit the GABA cells, we see a decrease. Same with enkephalin, and actually a little bit more of an intense effect. We see really nice seeking when they're just given vehicle. If we go ahead and inhibit these cells, we see a decrease. The glutamate data looks so good. It looks so promising, but it's a little underpowered, it looks like right now. In general, there's a trend toward if you inhibit CNO or you inhibit glutamate cells in the VP, we start to see an increase in seeking. So this kind of really nicely goes into the fact that the glutamate cells may be acting more as a brake on any type of seeking behavior. But again, nothing is significant at the moment, so we have to be a little cautious with that. But overall, what we're seeing is a nice complementing experiment to the stimulatory. If we turn things on, we enhance in GABA and enkephalin. If we remove them, we take away that seeking behavior. So it does appear that in the ventral pallidum, these GABAergic cells, specifically enkephalin, is necessary for cued seeking behavior. The glutamatergic cells, it seems that they're more acting as a global brake. Okay, so it's really great to be able to characterize what cellular phenotypes are doing and how they are, contributing to an overall behavioral state. But let's get mechanistic. Let's start to understand how these particular cell types are possibly resulting in these behaviors. Where are they projecting to? Who are they communicating with? How are we contributing to these different behavioral states? So what I'm going to now show you is looking at cell and pathway specific regulation from the VP to two other structures, the lateral hypothalamus and the subthalamic nucleus. And the way I do this is using an intersectional dread approach. So we're still using that dread, but now we're kind of fine tuning it a little bit. But before I show you the mechanisms that I use, let's just kind of look into why I even chose the regions I'm interested in. So Jasper, who again was a former grad student, now faculty at UAB in Alabama, he did a really nice study where he just put into the ventral pallidum an anterograde tracer. And he said, I want to look and see, he is a Cree-dependent tracer. Sorry, I just want to make sure that's clear. I'm going to look into these different brain regions and I just want to see what the terminal density looks like. So you can see in the GABA, if we look at the terminals in the LH, there's definitely terminals there. But if you look at them for the glutamate cells, much higher density. Converse, and why that's so important. So what do we know about the lateral hypothalamus or the LH? We know that it is part of the motivation circuitry and that it does mediate different aspects of addiction-related behavior. It's been very highly looked at. The LH is a really big hub for looking at food reward processing. But we do know it has a relationship to addiction-related behaviors. And what do we know specifically about this particular projection? We know that if we just look at the projection, not cell-specific, but just the general projection, it is important for feeding. But if we look at the GABAergic projection specifically to the LH, we know that it's needed for ethanol context-induced seeking behavior. So a specific type of seeking behavior for ethanol. So this was really exciting for us. It seems that there's definitely some involvement here. That's why we're going to look at the LH for one of our manipulations. The other manipulation we wanted to look at was to the subthalamic nucleus. So the subthalamic nucleus is a really fun structure. It is a very big structure that's looked at in the motor sensory world. It's actually clinically used. Deep brain stimulation of the subthalamic nucleus is used as a treatment for motor disorders, but it hasn't been looked at as much for motivational states or motivational disorders until honestly about the last decade. And the reason we were so interested in it is that look at this density of terminal fields from GABA and possibly some from glutamate, very marginal at best. But the reason we took these two areas is because we seem to have a really big difference. GABA is mostly projecting to the subthalamic nucleus, glutamate mostly to lateral hypothalamus. The subthalamic nucleus, we know there's some beautiful work where if you inhibit it, you decrease drug seeking, motivation, overall relapse. Similar to other areas, including the ventral pallidum, we see that it can have bidirectional control of natural and drug rewards. And that overall, if we're not being cell-specific, but just looking at the pathway, this pathway is necessary for ethanol context seeking again. So what were our hypotheses? My hypotheses were that if we selectively activate the glutamatergic projection to the lateral hypothalamus, we are going to decrease heroin cued seeking behavior. But if we go ahead and we stimulate this pathway, this is probably a pathway that is going to increase cued seeking behavior. So this is probably mechanistically how these different cell populations are contributing to their behavioral states. All right, so just to give you guys an idea, this is my timeline. This is the same type of timeline that I previously showed you. So I do the surgeries, the animals undergo self-administration, a forced abstinence period. I then do extinction training, I did not do extinction tests here. So before I showed you where I just went ahead and I stimulated those specific cell types late in extinction, I did not do that here. I just went right into the cued reinstatement test. But I just want to give you guys a little bit of idea of how did I accomplish this? How did I accomplish cell and circuit specific regulation? So we have our little diagram down here. So I have my transgenic rat lines. I have my GAT, which is GABA, glutamate, and I'm sorry, I don't have the enkephalin guys listed here, but I have my 3 different phenotypes that express Cre selectively in those 3 cells, all right? So we either have Cre being expressed or Cre is not expressed. What happens here is that I go ahead and I put my flip-dependent dread into my ventral pallidum. I put my Cre-dependent flip into either my LH or subthalamic nucleus. What does this mean? This means that if the flip comes in contact with a Cre-expressing cell, we will get an expressing dread. So we can isolate the cell and circuit specific pathway. If the flip comes in contact with a non-Cre expressing dread, or a non-Cre expressing cell rather, the dread never gets expressed. So this is how we are able to be cell and circuit specific. Okay, so what happens when we, for example, look at that VP to subthalamic nucleus pathway? All right, so here I'm just going to give you the end result so we don't have to go through all the behavior. What we see, which is really interesting, and because this was not our hypothesis at all, if we turn on this pathway, we actually decrease seeking behavior. So I always love getting data where it goes 100% against my hypothesis because I think it opens up more doors. But obviously it can make things a bit more confusing. But what happens then if we say, okay, what happens if we instead turn on the enkephalin cells going from the VP to subthalamic nucleus? Kind of a mixed bag. Not very conclusive. There's like no significance here. I previously told you that the subthalamic nucleus is an area clinically that's highly looked at for motor disorders and treatment. So the question that was really important to ask is, are we affecting locomotor behavior? Am I seeing this decrease purely because the animal is not able to move? The answer is no. So what I did is I did a locomotor test where we did a baseline session. The animals were given vehicle, which is that saline, and then CNO. And you can see that for when we activate this pathway, there's no difference in general locomotor behavior for saline versus CNO. And then just for posterity's sakes, we went ahead and did it for enkephalin too, and we also see that there is no effect. So it seems that what's happening here is that we are selectively decreasing the cued rewarding effects or motivational effects of the heroin paired cues when we turn on this particular pathway. This is really interesting to me, and this is something that I'm definitely in my own lab going to follow more up on because, again, this is counter to what our expectations were. All right, so let's look at the other end of the spectrum. What happens if we go ahead and activate that glutamatergic projection from the VP to the lateral hypothalamus? This is the one that I expected that when I activated it, I would decrease seeking behavior. And look at that. We got something that followed in line with the hypothesis, which is always nice. We had one little mouse where I'm not really sure what he was doing, but we do see overall when we turn on this particular pathway, we decrease cued seeking behavior. But there are some GABAergic projections here as well. So what's happening with the GABAergic projections here? Is it possible that maybe the GABA projection from the VP to the LH may be contributing to our seeking behavior? So do we have this like opposing cellular regulation within this particular circuit? We do not. We see that if we turn on the GABAergic projection, we don't see any differences in heroin cued seeking behavior. Again, this is interesting because for ethanol, contact seeking, this was a necessary pathway. For heroin, definitely does not appear to be a necessary pathway. So what is the seeking pathway? I was able to isolate pathways that are contributing to attenuation, but what's actually contributing to an increase in seeking pathway? And so we have our VP at the center of the universe right here, and there's a couple options that, we can look at. The main option is really maybe the feedback back to the nucleus accumbens. Could also go ahead and look at, now let's bring in a different neurotransmitter, the cholinergic projection to the prefrontal cortex or the BLA. We also know that the projection from the V to the VTA is important for cued seeking behavior, and that both glutamate and GABA project there and show opposing regulation. So this could be an interesting one to also go down. So kind of tons of options available to us, which is really exciting, and definitely some really, really interesting developments in the future for sure. Okay, so I know I don't have that much time for my last point, but we'll just get through it as best as we possibly can. And so what I want to change from now is I just gave you a lot of data that showed you how important the ventral pallidum is to heroin seeking behavior. I showed you data showing you that the cellular phenotype is important, but also where that cell is projecting to is really important. So this type of modeling is really fantastic. We need it. We need to understand the neurobiological states that are contributing to addiction, right? But what's also really important is to be able to say, are these particular mechanisms naturally in place or naturally contributing to vulnerability or resiliency? So for example, are GABAergic cells in a vulnerable animal naturally what's driving that behavior? And in a resilient animal, are glutamatergic cells what's naturally driving that resiliency? How can we look at this? The way we can really look at this is using a model that captures that variability. So let's go back to the beginning where I told you that not everyone who uses a drug ends up being diagnosed with a substance use disorder. Only about 8.5% of the population receive that diagnosis. So this is telling us there is an extreme amount of behavioral variability in the human population that can confer a diagnosis. We also know, don't read this. I know the instinct is to start reading, don't do it. We also know that there diagnostically is a lot of variability. This is just the DSM criteria that goes into someone getting a diagnosis of substance use disorder. And what I want to draw your attention to is the fact that in order to have a moderate diagnosis, you only need to have about half of the criteria for either moderate or severe. So even clinically, there's a recognition that everyone is different. You could have two people with a substance use disorder diagnosis that could have completely different criteria. So how do we capture that? How do we capture this variation in animal modeling? How do we take this complex behavioral states and distill it down into an animal model? Well, there's been some really beautiful, sorry, I'll only go to 11:50 and then I'll stop just because we started a few minutes late. So there's a few models that are really beautiful that do look at individual differences in substance use disorder. For example, if you're really interested in cue reward learning, we have the sign tracker goal tracker. If you're really interested in just like compulsive or taking behavior, high and low responder. Impulsivity, if you want to look at traits that maybe map in to substance use disorder. And then this particular study is a really beautiful study at this point, about 20 years old, but this is the study that really changed the perspective, I think, of behavioral neuropharmacologists. This was the first time a group said, this is from Piazza's group, that let's try and make something similar to what we see in humans. So they took 3 different behaviors, preclinically relevant behaviors, and then they scored them. And they then summated those scores. And so they did something a little more similar to what we see over here. So these models are wonderful. They're really powerful. I've used several of them. But just with any model, you have a caveat. And the caveat is these models all rely either on how one particular trait can predict vulnerability, or it's relying on linear relationships, which is what this does. Why is linearity a negative? Linearity can be a negative because I already told you, not every single individual has the exact same diagnostic profile. So how do we capture this type of behavior? We capture it by using a model where now we're looking at several behaviors across the different phases of addiction and using statistical modeling to now say, is an animal more in a vulnerable or resilient behavioral phenotype? So I'm not going to dwell too much on this. I just want you to be aware that what we did, and this is using rats, is we did self-administration and then we did various assays looking at motivational states and different seeking states. And so essentially what I did is I took the animals, and this is about 1,000 animals in this particular model, and we took behaviors from when they were consuming heroin, when they were in extinction, so they were trying to withhold, and then when they were in their seeking, so their relapse state. We took those behaviors and we applied what's called a connectivity plot. So what we essentially did is each animal is one of these colored nodes, and the lines are lines that are connecting each animal to its closest related animals. We then apply a clustering strategy. So now we want to ask the question of, normally when we do clustering in behavioral neuroscience, we look at linear relationships. We look at principal component analysis, k-means clustering, things like that. And what's the issue when you use linear analysis? You're essentially trying to find the projection that explains the most variance. So you're essentially just cutting through data in a linear manner. And what you end up doing is you end up creating a picture that looks pretty similar to what you started with. However, if you use nonlinear modeling, what we're now trying to take into account is how different community structures are interacting with one another. For example, this is more on the internal of the structure. This is on the external. This blue is closer in color and proximity to this blue than it is to this red. So when we use a nonlinear model, where now we're not just relying on this linearity, we see that we come up with something very different. And the reason why that is so powerful is because we are able to say, how do different behaviors interact in a nonlinear manner to come up with an overall behavioral phenotype? Let's see what exactly is contributing. Are animals maybe having specific taking patterns that are also related to extinction seeking? How are all these behaviors coming together? Let's get a little more complex here. And we also have stats on our side where we're able to also look at validation. So we're able to know, unlike k-means, how many clusters are we able to pull from this data? And we can validate it. We can say, how certain are we that animals fit into a cluster? And then how much do clusters differ from one another? So what happens when we do this? We end up having 3 behavioral phenotypes: vulnerable, intermediate, and resilient. And we see that if we look at their behaviors across the different, or their general behaviors, we see that our vulnerable animals overall are showing an increase in overall consumption and extinction and seeking, whereas our resilient animals are showing lower and intermediate in the middle. For all and I have about 2 more slides, guys, and then I'm done. For all the behaviorists out there, we did do extensive, testing to make sure that our model was valid. And this is actually all recently released in American Journal of Psychiatry. So compared to our resilient animals, our vulnerable animals show potentiated levels of heroin reacquisition, multiple relapse events. We see that akin to human populations, circulating ovarian hormones do contribute to differences in cued seeking behavior. When the animal is in a state where there's an adverse consequence, the vulnerable animals still take heroin. The resilient animals do not. We also see withdrawal-induced negative affective states in our vulnerable animals. And we actually, within this, are also capturing other models of individual variation, which is exciting. And then naturally, sex differences across all these behaviors. So why am I telling you all this? The reason I'm telling you all this is because if we do a gross neurobiological assay where we take our animals and we put them through extinction training and then a test for cued reinstatement, and then sacrifice them after that and look at CFOS levels, which is just a marker overall of neuronal excitability, and we correlate that across multiple brain regions, we end up with these connectivity plots. So over here, you'll see just the different areas that we quantified. If you don't see your favorite area, I'm really sorry. But essentially what I'm showing you here is vulnerable animals on top, resilient on the bottom. And so this is a little much to look at as is. And so I'm going to break these plots down into 3 different functional themes, if you will. And what I want you to really take note of is, first of all, the dark lines are unique connections within each phenotype. The light purple, those are shared connections. The ventral pallidum, shared connection between both of these phenotypes, which is really, really interesting. And again, as I'm emphasizing here, the ventral pallidum is involved in seeking and not seeking. It's also involved in extended amygdala stress. So this was really exciting to us because now in a model that captures individual variation, the ventral pallidum is coming online. And not only is coming online, but is a massive node. It's the most interconnected node for these guys. So this was really exciting to us, and I had some other VP data I was going to show you, but in the interest of time, I am going to stop there. I'll tell you that we have found some differences in ventral pallidal plasticity markers. We found some opposing regulation of microglia as well as extracellular matrix components for these two behavioral phenotypes, which is exciting. And where I'm currently moving toward is looking at using RNAscope technology. Let's dive more into this VP. Are we seeing that FOS is co-localizing more in GABA cells for these animals and glutamatergic cells for these animals? And then let's layer on top of it then looking at pathway specificity as well. So I'm really sorry I'm not able to show you the plasticity data. That's obviously my fault with my time management. But don't look at the screen. I'm going to skip forward to my thank you slide. Because I think that's really important to make sure all those people who did hard work with me are recognized. And so obviously my postdoc mentor in the lab and then our various collaborators across the country as well as the world. So thank you all so much. And if anyone has any questions, I'm more than happy to answer them at this point.
Thank you so much, Brittany, for this excellent presentation. Thank you.
Yeah, thank you. I'll go ahead and escape
So,
out of my presentation so that I can see people.
so let's open the floor for the question and answer session. Please feel free to share your question or ask it.
You know, I'm a very fast talker, so I think I just spew information. I don't even know how I'm actually very unaware of how I breathe half the time when I talk. And when I was pregnant, I was very well aware of the fact that I don't breathe as much.
No, it was very clear for me at least.
Good. Well,
So I can start with a question.
Sure.
So how we can define vulnerability and resilience in a neurobiologically aspects? Is it related to neuronal density, or like you mentioned at your last slide, is it the networking of these neurons? So how we can define these parameters?
Yeah, so I'm really sorry I was not able to show you that data. So the rat data is actually the bread and butter of what I focus on. So my main focus in my work is looking at individual variation in substance use disorder. And so as a trained behaviorist, my preference is always to use the model and to find the behavior that shows variation and then go into the neurobiology on top of it. Obviously, I showed you within the ventral pallidum, we have individual variability on a cellular phenotype of how cells are contributing. But when we use models of individual variation, we're now able to say that if I have animals that are showing more of a vulnerable phenotype and a resilient phenotype, now let's dive into the neurobiology. I showed you just really briefly, if we just look at overall neuronal activation and connectivity, we see gross differences between the two phenotypes. And then unfortunately, what I wasn't able to show you was the fact that we also have distinct neuroplastic mechanisms within the ventral pallidum as well as the prelimbic cortex that are contributing to these divergent phenotypes. Or I guess let's say are present in these divergent phenotypes because we haven't functionally tested them yet. So yeah, so if you want to look at variation, you need to first make sure you have the behavioral model that suits it and then dive deeper into it. Some of my more past work when I was in graduate school, I used the sign tracker goal tracker model, which looks at individual variation to cue reward learning. And so we saw there that there's some really marked and beautiful differences in neurocircuitry that's being utilized if an animal is attributing a motivational value to a cue or not attributing a motivational value to cue, just attributing a predictive value to the cue. So that's, as a trained behaviorist, I always go to behavior. And then once you have the behavior in the model, you dive into the neurobiology from there.
Okay, thank you. And I think it's a good strategy in research, in animal research, to go into clusterizing animals into vulnerable, resilient, different groups, since we have a complex variability in the human population. So to have this translation with animals, we usually use reduced number of animals for our experiments, and we don't systematically take into consideration this variability again in the animals. So it's a very interesting
Thank you. Yeah, I completely agree. And I think that's something that's really missing in substance use disorder research. I always think I saw Eric Nestler give a talk Oh my goodness, at this point, it probably had to be like 8 years ago when I was in graduate school. And you know, one of his main focuses on affective disorders, on like depression. He showed this beautiful network analysis at the genetic level that there are more genes being mobilized to maintain a resilient state than there is a vulnerable state. So I think that's something we really think about, especially in substance use disorders. We're so interested in the pathological state. We're so interested in what goes wrong. And we lose focus on, but what is in place to make sure it doesn't go wrong? And that's equally as important. And so I think that's why with modeling individual variation, it's so important, because it's not just about what's going awry that's resulting in this aberrant seeking behavior, what's in place that's making sure it doesn't happen. So GABA in VP, we have seeking, but glutamate is shutting it down. So it's just as important to understand the neuromechanisms that are promoting resiliency as it is vulnerability. Because in terms of treatment, if you only focus on one, you're missing a whole possible options of things, not just knowledge, but possible treatment options. That's just my big bias because I'm an individual variation person. But I think that that's a really important fact that a lot of people in addiction research don't account for.
Yeah, thank you. Other questions?
Yeah, if anyone watches a recording and has a question, you're more than welcome to email me or reach out to Neza, who sits 2 desks down from me right now, if you have any questions.
Okay. If no questions, I again extend my thanks to you, Brittany, for your presentation, for accepting to do the webinar. I
Thank
thank
you.
Sona and all people who attend. Thank you so much. Have a
Yeah,
great day,
thank you. I
everyone.
appreciate it.
Bye.